my distinct pleasure to introduce Dr. Zahra Shahrokh. Zahra has 25 years of experience in developing biotechnology products and have a track record of regulatory approval of 5 recombinant proteins, as well as additional clinical development activities. She's currently the Chief Development Officer at STC Biologics and a CMC consultant. Prior to that, she was the head of pharmaceutical and analytical development and also the head of CMC program management at Shire. Zahra also had leadership roles in Genentech pharmaceutical R&D development for 10 years. Zahra has a PhD in biophysics from UC Berkeley and other very impressive credentials. So welcome, Dr. Shahrokh.
So in this outline, I will be speaking to different modalities of submission, and I really want to give you a good sense of what are the different product development phases and what kind of regulatory interactions you want to have, what kind of package you want to put together to bring your product to the first IND. Really, the emphasis is on the first IND. I'm going to give you examples of gaps and deficiencies at the end, and would be delighted to hear questions, at the end. So let's see. Okay, so just some definitions. The IND application stands for Investigational New Drug Application. It's an FDA, basically a US form. There is, let's say if you're going to Europe, it's called CTA or Clinical Trial Application. And there is a format that now has become universal. It's a global format, which is actually called CTD, the Common Technical Document. And I'll speak to that a little bit and compare it to an IND format versus a CTA format. So these are the first applications into getting into clinical programs. Once you're ready to file, hopefully the product shows efficacy and safety, and you're ready to get commercial, the biological license application for biologics is called the BLA. Again, that's an FDA term. Compared to, for small molecules, you call them NDA, the new drug application. And if you're filing in Europe, it's actually called MAA, so the marketing authorization application. So actually, there are a lot of 3-letter words to memorize as related to regulatory submission. And then there are terms in what's called the quality section, which stands for the chemistry, for example, CMC, which stands for Chemistry Manufacturing Control. The F stands for drug substance. In biologics, that refers to, usually in small molecules, they refer to it as API, the active pharmaceutical ingredient. And then drug product is your final fill finish in the vial, as opposed to the bulk. Let's say the drug substance would be a bulk. So with these definitions, let's go to what are some of the different modalities, to file. So for example, when do you submit a BLA versus an NDA? So for biologics, anything that is a protein, virus, toxins, blood-derived products, gene therapy, even human tissue, those are all under a BLA, as opposed to anything which is synthetic, which would be NDA. And actually peptides fall under that as well. And then recent definition for what is a protein versus a peptide, I think the agency had, they had to be greater than, if I'm not mistaken, I think greater than 100 amino acids to be considered a protein. So this talk is really focused on, as I mentioned, IND, initial filings, and for therapeutic proteins. I won't be talking about these other modalities. Okay? So in this slide, I want to give you a good sense around what are the phases of product development, and again, with emphasis on biologics, and how and when you would have interactions with the regulatory authorities. Because again, in case you don't know, in order to put any drug, any compound into people, you have to have approval by the regulatory body of that country, in the US being FDA, and other countries have other names for that. And there are different types of information that you have to provide. So in the research phase, which I'm assuming most of you are in, you're really trying to figure out, find a target, that links is linked to some disease. You're trying to find a molecule that has some effect. And in a way, you know, you're actually doing like an in vitro or animal proof of concept. As at some point you identify what I'm calling a development candidate. So maybe you've screened different antibodies, you've screened different, you know, fusion proteins or whatever, and you say, this is my best candidate that I actually really want to take into the clinic with certain attributes. Could be it's EC50, could be it's, you know, behavior in PK maybe, or behavior in vivo. So typically before you set on to do toxicology studies, so at this point you would have done, again, in vitro studies, maybe some animal studies to understand some idea of what is the dose, you know, the dose response effect in animals. And you have to now start designing what I'm calling like IND-enabling activities, which is you have to show preclinical safety in some formal tox study, and that involves pharmacokinetics, pharmacodynamic studies. This is a good time to go to the FDA with what's called a Type C meeting and say, here's my program, here's a molecule I'm developing, this is the rationale, here's what I'm thinking about, you know, the clinical indication, and maybe my initial doses. So you have to have some idea of your phase 1 dose levels, and say, this is the tox plan, and really get buy-in to them. And I'll give you an example later on where not doing that has actually not been a good idea, because you can do your tox study, then go and have what's called a pre-IND meeting, which is you've done the production, you've done your tox study, and you go there and say, I'm ready to file an IND. And now the FDA says, well, this tox doesn't have the coverage. You can't do more than one injection, or, you know. So it's really good to have interaction with regulatory bodies and get their buy-in to your program. Then you file the initial IND. That allows you to go into the phase 1. By the way, I'm wondering, I'm moving my mouse. Could you see the cursor on the mouse be moved or no?
Yes, we can see it.
Oh, perfect. Okay, so then I can refer to specific. Okay, so you filed your IND to go into phase 1, and it's really your phase 1 clinical studies are targeted towards safety and getting really safety information. What is the dose that I can administer that is still safe and, you know, eventually would be efficacious when you look at later phases. And you get PK data because you want to understand exposure of the drug and how long it hangs around. And if you're lucky, and if you're in patients, obviously, you can get some pharmacodynamic markers and some, you know, biomarkers, a hint of bioactivity of that drug in people. Usually, there are interactions. You might be doing, you know, other changes, maybe manufacturing changes. There's an IND amendment to now go to your phase 2. You present data to the agency and then say, here's my rationale for the next phase, which is dose and regimen, the frequency studies. Now you're collecting more safety, but you're really doing what I'm calling here a human proof of concept. So while the earlier phases were animal proof of concept, now you're doing your human proof of concept. And that's key. It's usually by end of phase 2, you want to have collected, really have a good sense for that you have an active molecule, and now you're going to do your full-fledged, probably very, very large, you know, number of phase 3 clinical studies, which is collecting efficacy data now. This is what would give you the label claim, and you can file an application on. So ideally, you would have what's called an end of phase 2 meeting with the agency, where you present the data so far, you say, this is my phase 3 plan, but also all the way to market. You actually say, this is my CMC plan, this is my clinical plan, and how am I planning to file, what do you think? And really get good input from them. Now, if you notice, I have here what's called scientific advice, by the way, in Europe. It's a different, slightly different mechanism and different formality, but you do the same. You go to them and say, this is my plan, and get buy-in. And ideally, you get buy-in between different countries to do the same study. The most complicated is if you did it one study and only FDA approval, you know, you get only US approval, you can't get other approvals. So how you tie the different agencies together becomes more and more important as you reach this sort of phase 2, before phase 3 program. Then you have all your efficacy data, your safety data. You actually have good data to show. You would have your pre-BLA meeting and say, all right, I'm ready to now file my cert application, and this is my package, and and again get endorsement. They may have some questions. They say, before you file your license application, we need X, Y, and Z. And I've seen that happen. You know, it could be sometimes as much as you need to have a little bit more clinical data, or you need to have some assay added in there. You know, not very happy, surprised to have it at that point, but it could happen. And then, of course, aftermarket application, even if you get approval, there are always, I don't know of any program where you wouldn't have what's called post-marketing commitments, PMCs. Europeans call it follow-up measures, some. But you basically, the program doesn't end. It continues, and you're always doing more studies for collecting, from a clinical perspective, maybe collecting more immunogenicity data, or doing some label expansion studies, or you're making changes in manufacturing. Now you need to scale up, you know, to really meet the market demand. And so the program never ends. In fact, it explodes. You know, once you're, I would say in terms of resources, once you go kind of pretty much beyond phase 1, it's going to be like explosive, you know, goes up. So that's just an overview, right, of the development phases. I think I already went through this in terms of the clinical trials. Really, the most important aspect of your phase 1 is that your first in human is that you're really trying to get some sense of, is this, what's the lowest dose that is effective, and what's the highest dose that doesn't cause harm? It could be done in healthy volunteers, or in certain indications, or orphan drugs, or certain neurological indications. Obviously, you won't do that in healthy volunteers. You go directly to patients. And really, the important thing is you're assuring safety and the rights of subjects, and that ties to all the forms, the consent forms, and everything else that comes into a phase 1 trial. I won't move into the other trials, but just in the interest of time. Let's move to the next slide, which speaks to what do you file? You know, what are some of the differences between filing in the U.S. versus Europe? There could be certain advantages. There, for example, there are centers that maybe do certain studies in Europe that you may want to do a study there. But ultimately, you would have to file in a given country to get approval, you know, in that country. So the FDA is one agency. Basically, the reviewers are in the same agency. Europe is very complicated. They actually have many agencies. There is the European Medicinal Agency, EMA. It's really an administrative organization. They rely on scientific input from what's called CHMP, the Committee for Medicinal Products for Human Use. So that's where you go for scientific advice. You get input from multiple countries, and it's for all Europe. So it takes a little bit longer. There may be different practices, different cultures, but you get a collective input, and therefore when you file to EMA, you get a collective approval or lack of. So it's interesting where U.S. basically is sort of like a bottoms-up approach to, you know, to filings. It's European is sort of more top-down. There is, you know, they look at, you know, maybe more benefit risk of the whole package. You get more specific comments in the U.S. Ideally, you want to send, you can send the same package to both, and I've seen it, but you may have different outcomes. So that's the trick around product development to be able to manage, you know, global approval. In terms of forms, physical forms that you submit, I mentioned for the IND, it's an FDA Form 1571, which is basically the application, and it would have certain table of contents, introductory statements. You would have your investigator brochure, the protocol, then the CMC package, the Pharm Tox package, which is really your preclinical, and then if you have had any previous human experience at the end, which obviously in a phase 1 you won't. For a CTA, clinical trial application, let's say you go to UK first or Germany, they're similar. It's a different application form, but similar content. There, for the CMC, basically there's a protocol, an investigation brochure, but then you have what's called IMPD, the Investigational Medicinal Product Dossier that you file. But it's the same content. The quality is the CMC, the Pharm Tox is similar. There you have a little bit more risk-benefit. They want it early on. In the next slide, I'm showing the 5-review process. And by the way, these are all published. These are online. You can actually go to the FDA website and you can see documents that describe, you know, what happens. But typically, you file in the FDA. It goes to a division that is the reviewing division is going to be directed by the clinical indication. If you're in oncology, it'll be, by that office versus, if it's neurological. And what they do is that they bring in CMC folks, because those go across indications as necessary, or perhaps people from different divisions as necessary into that review body. And usually within 30 days after submitting an IND, basically you receive officially that they have received it, and if you don't hear any comments back, you're ready to go. In EU, you do have to actually receive, a form, and both the ethics committee and there's a national competent authority that has to approve it. So that takes a little bit longer before you can proceed. The content is pretty much the same. The data is the same. It's just maybe presented slightly differently. So let's now move into the more core, details. So what's in an IND? It's really 3 pieces of information. It's around your, as I mentioned, CMC, which is the chemistry, manufacturing, and controls. How did you make this product? How are you testing it? And you need to give a sense of assurance that what you're going to administer to people is safe and have some benefit, is bioactive enough to expose humans to. Then you have the non-clinical package, and I'm calling it non-clinical as opposed to preclinical. This is also a kind of a verbiage that distinguishes a little bit. Preclinical could be anything from, you're doing any kind of animal in vitro studies. Non-clinical implies that now you're doing like GLP studies, you're really doing your tox studies, and they're so-called IND-enabling. They're later stage, they're bigger, they're much more expensive, and they're very well documented. And you have to have those reports, before you can proceed. But the purpose is that do you actually have adequate, safety information in animals to support exposing the exposure levels that you're proposing in the clinical program to go into humans. And of course, the clinical area or package justifies why you're doing this, what is the background, and what is the clinical protocol, is it justified, and a lot of details related to how are you going to, what are inclusion criteria, exclusion criteria, and how are you going to escalate doses, and so forth. So to craft an IND package, it's really a story, and I want to emphasize that. It's not long, it's not data dump, but you're giving them assurance, as I mentioned, that you're not exposing the subjects to unjustified risk. And if you're doing studies in patients, obviously that they are going to benefit from it. Of course, if you're doing healthy volunteer studies, they don't benefit, but at least they're not harmed. In the CMC section, you're getting more into depth of how are you controlling, that material that you're making. What kind of controls do you have? Does it have appropriate purity, activity, stability? Are you dosing? Do you know how well you're dosing? You're dosing your molecule. As I mentioned in the non-clinical program, you have appropriate safety margin. Now that gets into so having higher enough levels that gives you safety margin in patients, usually minimally 10x. Ideally you dose in 10x is also what you're going to dose in patients. And there's a whole discussion around, what species and how many species and what kind of analyses do you do, from histopathology to clinical chemistry and so forth. In a way, you're trying to, as much as possible, mimic what you're going to do in humans and be able to predict potential safety concerns. So as mentioned, this is not a technical report, right? It needs to be pretty succinct and obviously accurate. I've helped clients where you read them and they are as simple as typos, which is not good, to maybe not necessarily or even miscalculations, so forth. So it's really your company, your organization, and your credibility. So it really pays to have multiple reviewers, and that's why when you're in a biotech company, there are a lot of people. You have way too many more people than you have in academics to just review things and make sure they've been stamped for quality. As I mentioned, tell a story, right, that is integrated. So in terms of timing, so thinking about it, is from basically go backwards from when you think you're going to file. Ideally, you want to allow yourself about 9 months to start preparing, and that means you have defined, who is going to start working with you, from be it consultants or partners or CROs. and who internally is going to be, sort of the contact person for a given discipline. Obviously, you have to read the guidances, really understand them. It takes at least 4 months, even when you have the whole team together, to put the information together, review it, and make sure that it makes sense. Now, highly recommend that you get people together from the different functional areas, about 4 months before that, and really have them talk. What I'm seeing as a big gap in again, more younger organizations that are doing this the first time is that clinical people are doing their own thing, and they don't know anything about CMC, and by the way, vice versa. And so there's disconnect. There are these islands working on their own, and if I can't emphasize how critical it is to bring them together, have them talk. The only place where you're forced, into having a conversation between CMC and clinical people is when you get into clinical administration. Okay, I need to put in an ID bag. Well, do you have stability data in the ID bag? Oops, I need to talk to my CMC people. And I've had that before, where it's a month before filing an IND. It's like, okay, this study is going to take 3 months. So I'm sorry, you didn't talk to me about that. So I can't emphasize more about integration and bringing teams together up front and jointly planning. Then each person goes and does their thing. Then you come back and kind of say, okay, here's my story. How did it fit into the rest of the story? Defining who's responsible for, for reviewing across all sections is also important. Once you write it, it's really, it's tricky where, and I'm sure the regulators and the reviewers have this issue where it's obvious, 10 different people wrote this. So the style changes, it becomes really confusing, the level of detail. So ideally you have, again, either people really trained or working with your scientists to describe how much detail and what kind of information you put in there. So it's not like it's, 20 different books in one. And then, of course, there are annual updates. You show more and more information as you proceed with the program. So in terms of format, I mentioned there's the IND format, but this CTD format, highly recommended to be used because definitely as you get to later stages for commercial approval, that's what you need to be, that format is what you need to be in. And I have here some websites. At the end, I also have a whole bunch of other references where you can go and read about them. But this triangle shows very nicely the buckets. As I mentioned, there are 3 buckets, right? There's what's called quality, which is the CMC module 3, the non-clinical, the tox studies, it's called module 4, and the clinical program is called module 5. And there are layers. So at the bottom of it is all the details. Then usually for phase 1, that's all you do. You have your modules 3, 4, and 5. Module 2 is when you start doing quality, you start doing overall summaries. So if your module 3, which is the CMC section, is, let's say it's, I don't know, 1,000 pages, maybe you have, 20 pages of a quality overall summary. I've typically done that by phase 3/commercial. So it's just, I just want you to know that this is the organization. As you get later and later stages, you have to provide more summaries and rely on the body, of the details that you provided before. You're building on that as you go further into clinical development. And I'm going to go through what is in each of these sections and modules. In this slide, I'm on slide 15 now, I'm comparing side by side, what's in an IND versus a CTD. By the way, the CTD can be submitted in a electronic format, so it's called eCTD. And there you have to put additional, you get your IT involved heavily. You have to have additional ways of reviewing and connecting. For example, linking, you click on one section, it links to the other section. It's very nice. It's very, very convenient for the reviewer. A little bit more work on the part of the sponsor who has to put it together. But basically, as I mentioned, both of them, of course, you need the protocol, you need an investigational plan. It's just that for the CTD, it goes into different places. Like module 1 would have some of your, your investigational plans. As I mentioned, CMC section is called module 3 and non-clinical is module 4. And if you have any previous human experience, it's going to go into the clinical reports in module 5. So similar content, a little different format. So the introductory statement or the introductory section is really around, this is where you're basically providing your perspective to the FDA. What are they expecting? What is your development plan? Specifically, if it's phase 1, you're going to talk about, How are you going to, what kind of monitoring you're going to have for human subjects, and what you're going to look for. It's really your plan. What data are you going to collect? And it's very brief, 2 to 3 pages. Then you get into I'm going to go a little bit deep into the CMC section. That's my core expertise, the CMC section. They are divided by what's called the S section, which is for drug substance, and P section for drug product. So you see here 3QF is basically module 3. And section 2 in here actually refers to a format, which is the CTA format, but the S stands for drug substance. What's common between the drug substance and drug product elements, and just a reminder, drug substance is your bulk material. You've made it, you've gone to a 200-liter scale, you've made your bulk, typically you freeze that away, hopefully, and then whenever you're ready to fill the vials, the actual final vials or pre-filled syringes or however you plan to administer, that's your drug product, that's your sterile product at the end. So those are completely different sections. In both of them, what's common is that you have sections that refer to who's the manufacturer, where is the testing site, who's doing stability program. Then you describe them, it's called manufacturing process description. You describe what are your in-process controls. For example, to go from one column to the other, what do you test for? You show how you're controlling the raw materials. Very important. What you bring in, is it, for example, very key is it from animal or, animal source or not? If it is animal source, you have to absolutely have proof that it's, that it's BSE, TSE free, right? It doesn't have the encephalitis, viruses. And ideally everybody goes to animal component free. But you have to describe what your raw materials are going into the manufacturing process and how you're controlling them, as well as how you're controlling the drug itself. What are your specifications, what assays, what criteria? Then both sections would have batch analysis data. Here's the data on 1 or 2 or 3 batches you made, and of course stability data and summary. What's unique is that the drug substance talks about actually your whole elucidation structure. What is the structure of your molecule? What are the impurities? What kind of forced degradation studies you've done? The drug product emphasizes formulation development, how you came up with that, and compatibility, let's say if you're in IV bag, all your in-use studies. So it's a later stage section. So there are some differences between them. In the manufacturing process itself, whether it's drug substance or drug product, as I mentioned, the left side, this characterization section is really belonging only to the drug substance. But the right is very similar, whether it's drug substance or drug product. So in the characterization section, this is where, again, for biologics, you're showing every detail that you know the sequence well, you understand something about the folding of the protein, you've done perhaps other biophysical characterization. Then the size, charge, heterogeneity, very important to describe that you have some methods for determining the heterogeneity of whatever material you're making. And the FDA looks at this point, most agencies are looking for orthogonal methods. For example, different ways of looking at molecular weight. You could look at it by mass spec, you could look at it by molecular weight markers on a size exclusion HPLC column, you could look at it with molecular markers on SPS page. And they each tell you different aspects of the molecular weight. And this is where you would throw in as much as you can, information around characterization. Obviously, at the beginning, it's a small package, maybe, only a few pages. But as you get later and later, then there's expectation that you're going to know more and more. What's really important out of all this, which I have here in magenta, is that somehow from all of this, you're defining what are my critical quality attributes. That's the basis for your specifications, which goes into the controls, which I'll come back to in the next section. Let's say, for example, we know that there's a concern around aggregates and immunogenicity. So you call aggregates as a critical attribute, and you'll say, I'm going to measure it with size exclusion, and I'm going to have a specification, a range around aggregates. So that's how this section links to your analytical procedures for release of the product and specification. Then it goes into impurities. Obviously a big issue, right? What is happening, what is coming through from your host, right? From DNA to proteins to leachates from columns. So all of what's called process-related impurities. as opposed to what are the product-related impurities. So we talked about aggregates, maybe you have fragments, maybe you have deamidation, oxidation, other degradation products. If you are dealing with conjugated materials, you know, how much is unconjugated, what's the level of, let's say you're doing pegylation, how much free peg you have, what is everything else that is coming from your process into the program. These are your, into the final product, these are your impurities that you need to describe in its own section. You describe these are potential impurities, and these are the assays that I have to actually go and detect them, and this is how I'm going to control them. So, for example, either control could be that I have an assay that at the end that says I cleared that, whatever, I had BSA in my process and I cleared it, or a control could be that, you know I have a small molecule and I have a USDS process, and as long as, you know, the millisiemens, you know, whatever conductivity reaches a certain level, I know I got rid of those, for example, salts or small molecules. So it depends how you describe your what's called control strategy. Thinking about it early is really important because it helps you then to have a seamless program going through and to have less of a kind of headache with agencies. So that's the section. For drug product, now you need to show in going from, let's say you've frozen it, your drug substance, now you thawed it, and then you fill it, you know, filter it and fill it into the final vial, and then it's sitting in the vial, it's sitting with a stopper, it's going to be stored. What potential impurities can come through, you know, from drug substance to drug product. On the right side, I mentioned we have to describe the manufacturer, you describe your process flow diagram. Here you have to start talking about what is a critical step. For example, if it's an antibody, you have a protein A. That's a critical, basically that's a critical process step because it's actually cleaning, it's doing your major cleaning. It's an affinity purification, it's major cleaning step. And then you would talk about how are you controlling those critical steps. It could be that, you know, it's the way that I elute the column. It could be the way I'm, you know, controlling the conductivity, or I'm having these in-process assays to tell me that I'm, you know, getting rid of wholesale protein, for example. So this section describes your manufacturing process flow and how you control them. Then you need to have sections that says how do you come about that. So your whole development history, especially with the cell line, you know, what was the design, you know, around the vectors and the cell line, how did you make it, all the viral testing around it needs to be shown to, again, to show safety. How do you come up with downstream process, with the purification process, and if you did changes, I've had cases where the tox material was made with slightly different process than your clinical material. Now, almost right up there, you have to show that I have comparability of product quality between tox and clinical, because otherwise you can't link that tox program to your clinical program. And of course, in the drug product, the whole emphasis is on aseptic processing. You know, how well you're controlling and you're having a sterile product. So tied to the other sections, which is your analytical procedures, specifications, and batch analysis, so these are the rest of the CMC section, they're also, as I mentioned, tied to your characterization section. So you select a subset of characterization assays that are tied to your critical quality attributes. You call them release tests. Some of them are given, like compendial tests, like you have to do, for example, you know, for final product you have to do sterility. It has nothing to do with whether it's a small molecule, a peptide, a protein, a gene. It has to be sterile before you inject them to people. So those are like compendial release tests you have to have. But others are a subset of characterization tests. Then, as I mentioned, you define specifications. And here, let me also, my pet peeve to clarify, specs are a combination of what assays you have and what the acceptance criteria are. In other industries, you refer to specs actually as your criteria. In our field, in an IND setting, your specs are the combination of the assay and the criteria. Obviously, a criteria is tied to an assay. So you want to be very clear that refer to acceptance criteria when you're talking about my aggregate has to be less than 5%. That's an acceptance criteria. The spec says I'm doing this by SEC and have an acceptance criteria of less than 5%. And then there's a section that justifies. So where did you come up with 5%? You know, how do you know that's good? And it's based on a combination. Obviously, early on, you don't have human data, so you're going to tie it to here are the assays, this is my analytical capability. You know, an assay has a precision of whatever. If it's 5%, I can't have a spec that is less than that. What about manufacturing? Is something changing on stability? So, for example, I've had a case where you could have a change in, let's say, even aggregates over time, or let's say deamidation over time. You would set a much tighter specification at release, at T0, right, at baseline, compared to your end of shelf life. You could do that. It's in European language, it's actually end of shelf life specification is very obvious. In the U.S. terms, you have to define it a little bit differently. You know, you basically are setting in a way tighter, internal or drug substance spec than your actual, final product spec. And then the other section that ties to this is all the batch analysis. So the more runs you've done that are representative and you can show all the data, the greater rationale you have for justifying the specs. You actually have data to justify it. And in the phase 1, you may not have much. So you're relying on history of maybe other programs like that. If it's antibodies, there's a lot of knowledge out there and a lot of history. If you're dealing with something very new, it's a new cytokine, then you try to tie it to maybe closely related family members, or really have to justify it by experiments you've done. I'll give you an example. One of the things that I work with a lot with clients is you don't know. Let's say you've done your charge. You have let's say a capillary electrophoresis assay, or you have an ion exchange assay that shows you have 20 different peaks. What do you do with that? How would you set a spec? Well, it's very simple to collect those fractions and do a very simple thing. Characterize them. What are they? Is it deamidation? Is it oxidation? Or put them in bioassays and see if it's active or not. Goes along with just saying, look, if it's active, I'm a little less worried about it than if it's something that is dead or potentially immunogenic. Now you want to put actually a tighter spec around it. In the next slide, I'm emphasizing then the sections that are mostly tied towards your drug product, which is the formulation and stability sections. So here you define some of the pre-formulation work, which is really your biophysical, biochemical characterization of, how does this molecule degrade? What do you understand about it? Does the structure change on pH, on, say, buffer? So basically, it's the basis for rationalization of your formulation. Why did you pick a particular pH? Why did you pick a particular buffer or excipient? And the protein concentration itself often is to support a clinical dosing. So there is a reason, you chose these parameters. This is the section you justify that. Force degradation studies are done actually relatively early. These are also important things that sometimes get missed by people who do not have developed products before. And I'm including actually some of the CMOs, the contract manufacturers you may go to. So highly recommend that you have a good CMC person on your team that actually manages that. But you want to understand, is this product susceptible to light, to perhaps oxidants that you might have, certain things that might leach in the process. I had a program where, and it took about a year, by the way, to resolve, and it was very late stage in the program. On the SEC, on the size exclusion chromatography, I would see a peak that looked like it was very, very large, many, many, many multimers. And it showed absorbance at 214 and at 280, which suggested there's protein in there. But for the life of us, we could not characterize this. It turned out that it was a absorption of some of the protein to the leachate from the drug substance container that took about 6 months to form. It was polymeric. It was a polymer that was taking 6 months. This peak would show up after 6 months. I would never see it at the beginning. But it was showing up, and of course there was an FDA question, go and figure this out, and we had to go and change some things to actually minimize that leachate, and the peak never showed up again. But that took a year. So hopefully, these are not usually your problems at the beginning, in a phase 1, but just some things to be aware of. The more you understand your program, the less problem and headache you have later on. You have to justify, speaking of leachates and extractables, you have to justify the use of a particular container closure. You actually do what's called a risk assessment at this stage. What process you're using, what potential leachates could be there, and are you showing their clearance? Later on, you actually physically have to have assays to show what kind of leachates came out. The non-clinical, so just switching. So this was on the CMC front. Switching to the non-clinical section, This is where you're going to be doing pharmacokinetics and toxicology studies using the same schedule, duration, formulation, and route of administration as you propose in the clinic. And basically, minimally, you need to have data to support the starting dose. And obviously, ideally also, you know, as I said, safety factors above the dose that you're going to go, highest dose you're going to go into. So there is a section around pharmacology, which talks about mechanisms of action. Is there cross-react? This is really key. What are the cross-reactivity with, you know, tissues, with of the drug with human tissue, and therefore justifying the animal tissue that you picked, because it needs to represent, for example, if you don't have cross-reactivity, let's say if this drug is supposed to be picked up by, I don't know, hepatocytes, so your concern might be, safety concern might be around, you know, liver toxicity. If the animal model you use in the tox study, that drug is not taken up by the animal hepatocytes, that tox species is not a representative tox species. So the data would not be useful in predicting human potential liver toxicity. So that's what you're doing in this whole section in the pharmacology studies, and the reports go in there. Bioanalytical, I mentioned to you, you have to have PK, pharmacokinetic assays. You have to have actually assays around immunogenicity for biologics. You always have to have an immunogenicity assay. And you're doing those studies also in animals. You're showing the PK in animals. You're showing if there's drug interaction, if you're putting three different things in the animal. And you're, again, justifying the species. And in fact, you do multiple species, and then you do species scaling, right? So there are different scaling models to say from between a mouse, a rat, and maybe monkey and human. You can predict this could be the PK, this could be the exposure level, and based on that you design your tox program, which it's anywhere from your single dose acute dose to more. If you are dosing humans more than once, then you have to have multiple repeat doses. For example, minimally a month if you're going to do more than, let's say 2 or 3 doses in humans, and maybe 6 months if you're going to do more chronic, as you do with, many, for example, orphan drug indications. You have to dose them if they're kids, pretty much their lifespan. Genotox and carcinogenicity, those are typically not for biologics. Reprodox and developmental toxicology, again, for small molecules, you have to do them up front. For biologics, usually they're done a little bit later. It would have to be something pretty unusual. Maybe you have an immunotoxin. Obviously, then now you have to do that earlier on. And then local tolerance, if you are doing anything other than IV. You're delivering sub-Q, now you want to actually collect information around the local inflammation, site of injection reaction, and so forth. And as I mentioned, immunogenicity for proteins. The clinical section, you have to have data on what's called pharmaceutical studies. This actually, again, most of this relates to, for small molecules, is your obvious, oral availability, linking your in vitro dissolution test in vivo. But in the context of proteins and injectables, this could be, for example, you're going to be doing if this is biosimilar, you're doing a bioequivalence, comparison of the originator with your biosimilar, for example. What do you know about how this drug is going to be administered to patients? Then the PK, I mentioned, pharmacodynamics, PD, and anything that you've done on safety and efficacy on humans. Of course, this section will be very small. If you don't have human experience, it'll be mostly your protocol and investigator brochure, and the PK studies, or PK design and clinical design. Then there is a very important link between, in this next slide I'm showing link between the non-clinical and clinical section. So you use the tissue cross-reactivity, as I mentioned, to justify your non-clinical tox, which then justifies your, the dose and safety margins in humans. Your pharmacology studies, for example, plasma binding, are there shed antigens, how is it being metabolized? All of that tie into both PK and pharmacodynamics in humans. So there's a lot of linkages here, and in the IND, you want to make that link. That's what I'm talking about, a story. That's how you build the story and rationalize a certain data impacting. You're really looking for impact of data on other on other aspects of your product, be it CMC impact on non-clinical, CMC impact on clinical, non-clinical impacting clinical, and so forth. And now I want to switch gears and talk a little bit about some of the potential deficiencies that you might see, or I would say missteps towards an IND, or even actually during development. So I mentioned cases where the tox study was really not adequately covering the clinical dosing and regimen. And again, this is where I highly recommend go and talk to the FDA, say this is your plan, and justify it before you spend millions of dollars. Actually, animal experiments are more expensive than humans. Monkey costs more than humans. So, so make sure that you've had that conversation before. One of the things I've seen is missing, so for example, people may have missed developing anti-drug antibody assay, or the bioanalytical assays are not validated. This is one case where before actually using the assays, these have to be validated. For CMC work, when you're developing, let's say, a release test, in an early clinical program, you don't have to validate it. You kind of what's called qualify it. It's a little bit smaller scope. But for bioanalytical assays, they have to be validated. And if you don't know what that means, I've put some there's draft guidance, obviously, but there's also a really good article. I put that down here. Even though this article is about actually anti-host cell protein antibodies, it's very well written for perhaps even somebody coming new into it in terms of what do you look for? How do you even develop an anti-anything, antibody assay for human testing? So the reason I put in here the residual anti-host cell protein assays is that the Office of Blood Products, so if you're developing, coagulation factors or blood products, they actually require you to do this. Other divisions don't. Europeans like to see it, but they may not emphasize it at the beginning. Eventually, they may ask you. And one company literally, and I was they were unfortunate enough to, ended up actually when I got hired, it was because they had issues, and within a year they went bankrupt. And it was because all of their development work. This was when they were actually submitting the BLA, and they did not get approval. By the time they fixed the problem, they went bankrupt, and it was because throughout the whole development, they didn't have a good assay to determine that you actually have thousands and thousands of ppm of host cell protein that was actually generating antibodies in humans, and the process was not developed to remove those impurities. It's really a shame. It's kind of a sad story. But so make sure that you have good CMC people, that they are developing good assays, they're justified, so your process has adequate impurity removal, so you don't have those issues later on in the clinic and eventually in the whole entire program. Another example is, and this one speaks to the sort of the disconnect between what I call R, which is the research group, and D, which is the development group. And this is an example where in research a very promising anti-cancer cytokine was developed. There were different variants of that in late-stage research. They picked one to go into the clinic with, but the data was very inconsistent. It was highly variable in the animal models. And actually, I was involved in that program, and I got, this is many, many years ago, goes back a million years, but I started talking to research folks, and I realized they're putting this in an LSAT mini pump. So the protein is sitting in PBS at 37 degrees for 20 days, and it was precipitating, it was aggregating, it was not good. So we started doing some pH stability studies and found that this needs to be at a lower pH, and of course saved the program. New formulation came up, we have a great program, and it got, it can support IND-enabling. This interesting program actually later on developed toxicity, and it turned out that we knew from some biophysical studies that there is an interaction in that particular animal species of the receptor with the cytokine, and our tox model was not a good model. So we actually, or I should say, the human protein delivered into the animal developed antibodies and developed specific toxicity. So we made the monkey protein and did the tox study. It was clean, and the program proceeded. Otherwise, the program was going to be dead. So there are interesting things that you really want to bring in your formulation, analytical people into your, preclinical programs as early as possible, so you don't miss something. I just got a note that I have less than 2 minutes to finish, so I'm going to speak a little faster. I have a couple of more examples. Very common thing that I see, drug in PBS. Everybody uses PBS. It's wonderful for biology and for cells, but highly warn you that if you're dealing with a protein, usually you aggregate in PPS if you freeze it. And the reason is that the dibasic form of the phosphate actually precipitates on freezing. The pH drops by 3 to 4 units, and many persons are not happy at that low pH. So do your formulation development, make sure that whatever you're developing actually, do the freeze-thaw cycles and look for aggregation. I've seen proteins showing up as particulates later on in IV bag. You thought everything is great, now you're ready to file an IND, you do your study in an IV bag, holy cow, there are all these showers of precipitates coming along. The eye is very important. Get your formulation people involved, or even if you have somebody preclinical doing formulation, tell them to look very closely and do the studies early on, because now you get delays. Another example is this was a case of an enzyme. It was a very interesting molecule. It showed efficacy in animals. We moved to IND-enabling studies. The process development folks started developing the clinical candidate, and it was not efficacious. So big surprise, it wasn't even as soluble as what was done in the research group. A lot of back and forth, of course, a lot of, sometimes you point fingers and says, well, you guys don't know how to make a protein. Anyway, it turned out that in preclinical development, the mouse enzyme was used to show efficacy. And of course, you're developing the human enzyme when it became a development candidate, and process development was doing the human enzyme. And the two are very different. We started doing, the glycosylation was different, the charge variants were different, the solubility was different. So your animal model and what you did in preclinical had no relevance to humans. So of course the program died, and then later on, had to be revived many years later when there was more money to actually go back and fix it. But again, unfortunately, 1.5 years and $1.5 million spent, and the program died. So very important, understanding structure function, having the right molecule in preclinical development that you're taking later on into clinical development. And this is my concluding slide. Really, remove the disconnects, bring teams together, integrate them, really have good analytical people, and good formulation. It's sort of it's lost because nobody knows about that. I didn't know what formulation meant when I came from academic to industry 25 years ago. It turned out to be really a good skill to have, and just watch for it. And watch out for it. You don't have to do everything on the face of the sun. There's only so much money to get into phase 1 development, but sometimes quick and dirty really bites back and delays the program later on. So again, really needs to be more, how should I say, with a critical view, really understand what you're not doing. It's okay to say, I'm not going to do X, Y, and Z, but understand the implications and be able to manage. And I think that's it. The rest are all kinds of details that you can, again, the slides will be available. And at the end, I have, I refer to the Federal Code of Federal Regulations and guidances in both EMA, Europe, and FDA. Thank you.
Great. Thank you very much, Zahra, for this very nice presentation. I think we have a few questions, and Linda is going to read it. And for those participants who can stay on, we'll just go over a couple questions. And if you can stay, Zahra, to answer them, that would be very much appreciated.
Absolutely. I'm afraid. Yeah.
So I think one of the questions we have, Zahra, could you maybe define what a Type C meeting is a little bit more? Does that refer more to a pre-IND or a pre-IND?
Right. So it's different from the pre-IND meeting. So basically, if you have any questions, and it could be about your clinical program, it could be, for example, I have a gene therapy product, or let me give you an example. I was working with a company who was working on mRNA. Well, what does that fit into? It's not gene, it's not protein, it's not, if you don't know something, obviously go and talk to the FDA, but bring a program and actually have a meeting with them to say, this is how I'm planning to go and proceed, and get their input. So it's something that you can do earlier on. The pre-IND meeting, you can only have one. Type C meeting, I think you can request, well, hopefully not for the same topic, but you can request many of them. Pre-IND is when you're ready, you really are ready to, you've done most of the work, you're ready to file. And now you're getting alignment that, look, here's my clinical program, actually. And I have my CMC section, these are my specs, these are my package, I'm ready to go. I've done the tox studies, this is my tox, and the tox you have your tox data and rationalizing your clinical program, and there you're really getting endorsement around the clinical program, and then you're filing the IND. So there are little different purposes, and of course there are different meetings.
Another question is, what are the preclinical data and manufacturing facility requirements that you need to have in place to have a pre-IND meeting with the FDA? So specifically, this person is asking, would they need to have their GMP facility ready prior to a pre-IND meeting and IND filing?
Okay. Yeah, very good question. Okay, and I would answer, by the way, like the FDA does, it depends. Most programs, you ideally want to have made, you want to have made your protein the way that you think you're going to do a GMP. You don't have to have made your GMP batch, but you want to say, I know how to make it, and I have confidence. You want to have the pre-IND, because you can only have one pre-IND meeting, before filing the IND, you really want to have confidence that my manufacturing is covered, my tox program is good, and I'm going there to really get the endorsement around the clinical program and move forward. I wouldn't use the pre-IND to say, oh, I'm really trying to learn what I'm supposed to do. You see what I mean? That's where you would use maybe more type C, or actually start with consultants, get as much information as you can before going to and talking to the agency. The pre-IND should be like you're pretty close, but you don't have to have your GMP data.
And thank you, Zahra. And another question is, for an orphan drug, does that change this process?
Right, and I've dealt with many orphan drugs. The overall process doesn't change. What does change in a nice way is that typically you are doing a phase 1-2 and then a phase 3. So you're much more, much less clinical phases, but what that means now is you really actually have to be a little bit more ready up front. You have limited patients, you may be making only one batch, so it's a huge challenge around how do I know if I have a consistent process, how do I set the specs, what kind of impurity profile do I have that actually went into the clinic that later on I can justify commercialization. And ideally by phase 1-2, by the time you're in phase 1, you're developing your commercial program. You're not developing a phase 2 program because there's no phase 2. So my entire year at Shire, that's what we did. We had only a 2-phase program. So run, develop a phase 1-2, put it in the clinic. You haven't even dosed the patient on developing a commercial program. Obviously, with the intent that with planning for success, because otherwise you're going to be delayed on the CMC front, huge in a big time. You won't be ready because what you want to do, the phase 3 material better be your commercial program. So if you have a 1-year phase 1-2, that's the year you have to develop your commercial phase 3 process to then make the phase 3 material, have an end of phase 2 meeting with the agency with the phase 1 data and say, this is my phase 3 and commercial program. It's very squished, all the timing is squished tighter.
Thank you, Zahra. Another question is, how does a 505B route impact the IND scope? Right.
So we're talking about now generics, or in the case of biosimilars. That's a whole other strategy. Now, if you're talking FDA, which is a little bit different than EMA, let me just speak to EMA because they've been Europeans because they've been doing it for many years. They expect a phase 1 PK bioequivalence, and you do your phase 3, kind of slash some sort of a, in vivo human clinical trial activity, and that's what they require. There's no way around it. FDA has been the new guidance document has been actually rather creative. So creative and yet difficult in the sense that they actually expect that you come in, if you're going to have your first face-to-face meeting with them to tell them this is your biosimilar program, they want you to have made your commercial scale material, have that data, show that comparability, package the preclinical, basically the in vitro comparability, and come with a program. So you're now, spending a ton more upfront to be ready to go and meet. The package and information is still the same. It's just, for all of these, by the way, for all these programs, you still have to have a safe product, a stable product, right? And it has to be active and effective. But it's when you package it and how you package it and when you go, with what information to the agency. So I hope that answered the question.
Mm-hmm. And I think we have some other questions, but they're all related to cell therapy and stem cells. And Zahra, since your expertise is more in the protein realm, I'm thinking that, and we're running out of time here, I think it would be best maybe if we put those on hold, and obviously it indicates to us that we should do a cell therapy webinar to maybe address those. I think that might be more appropriate.
We could do that. If there is, if you want to refer them later to me, and I can, if I know them, I can respond to the people who requested it. I have no problem doing that.
Great. Well, that would be good too. So maybe what we'll do then is we'll forward you some of these specific questions related to the cell therapies, and then if you can respond to them, that would be really helpful.
Sure. Absolutely.
Great. Okay. I think maybe one last question is, what is the typical time requirement for doing IND-enabling studies and filing the IND? How many years does that take?
Okay. So yes, it's a very good question. Depends on where you are. So let's start from, do you have a molecule that actually, and let's say this could be a mouse version or something, right, that you've actually shown animal proof of concept, so you've shown some efficacy. Now you're going to go and do cell line development. So I'm going to tell you, because that's the longest lead to actually activity. So from the time you say, I have a molecule, by the way, but you need to have the human version. So I have a sequence. Let's say you have a sequence, now you want to make it. So it's about 6 months, right, to develop your cell line and, you know, and basically be able to start even process development. It's about another 6 months to have done all your assays and process and formulation. So I would say it's about a year of CMC. At the same time, obviously, you're doing your, you know, let's say eight months or so into it, now you have material to do your tox study. So typical, if it's very organized, you're really in good shape, I would say 18 months from the time you trigger and say, I'm ready, you know, I have a concept and I have a sequence to go. So that's, yeah, so that's the length I would allow. And probably about 5 million, you know. So.
All right. With that, I'd like to thank you again, Zahra, for the very informative presentation, and thanks everyone for participating, and stay tuned for future webinars. Thank you and take care.
Yeah, thank you very much. Thank you. Bye-bye. Bye-bye.